Thank you. David, thank you so much for inviting me to come to this meeting. It's incredibly meaningful for me. I work at the Arts and Science campuses of Columbia University. No translational research unless you can reach out to clinicians, and we try to do that, and I have collaborations, but coming face to face with the people who are affected by the gene that we work on is incredibly motivating. I don't think you could understand how motivating it is. That said, this is going to be a pretty basic talk. And so what I'd like to first do is just do something that you've seen many times before. P53 is like it has tentacles. It can reach out and affect so many different processes in cells. You can see the list here, and every time somebody made a discovery, like P53 causes cell death, P53 causes cell cycle arrest, that seemed to give us the clue as to how it functions as a tumor suppressor, and we still haven't the faintest idea how it actually works as a tumor suppressor, because invariably somebody else comes along and says, oh, I've made mice that can't do this, that, or the other thing, and they don't get tumors. So something's missing. And this keeps us all working hard in the laboratory. What I believe is that what P53 does is very context dependent. We have to look in different cells and different tissues and in different kinds of model systems till we can really get a full picture. We know that P53 is very good. It causes tumor suppression. It plays roles in modulation stem cells. You heard beautiful talk from Varda and also from Ihor about this. It even plays a role in fertility. But too much of P53 can be a bad thing too. And I'm going to show you a couple of slides to show you why that is. It's true that there are many families with mutations in P53 in their genomes, and this is the theme of this meeting and the outcomes of that. But it turns out that there's a very small, basically an N of 2 families that actually also have mutations in a gene that regulates P53. And in fact, fascinatingly, and it's, of course tragically as well, it predisposes it to another kind of syndrome, which is also potentially lethal, which is premature aging. It's progeria. And I won't go into, because I don't have the time, this is to discuss this, but having too much P53 can be a bad thing. And so it turned out that colleagues in Germany, for a number of reasons, human geneticists discovered a patient. They did the analysis, and it turned out that they have a germline mutation in MDM2, which is the negative regulator of P53. And this just shows the bottom line over there. Is there a pointer here or something? Oh, this works, haha, okay. So right over here, this extends MDM2 a little bit. It makes the MDM2, and very briefly, basically unable to degrade P53. And you can see you've got extremely high levels of P53 in cells or cells from this patient. And we've been working with our colleagues in Germany trying to understand how this mutation works. So I thought I'd just put this in because it's essentially the other side of the coin. Now I'll come back to the bad things about P53 that are relevant to cancer, and just remind you that P53 is mutated with extremely high frequency in many different kinds of cancer, and in many and it's much more than most of the other known cancer genes. So this is why we have the impetus to study P53 because of families like you and because of general sporadic tumors as well, where P53 mutation is quite common. And as you've seen this before, most of the missense mutations, the mutations that actually cause these full-length proteins that wreak havoc in some cells sometimes, are located in the DNA binding domain. But one of the things I want you to think about as you think in the future is that it's not only that they're adding oncogenic activities to cells. Losing wild-type P53 is a very important part of the question. But we'll start first with some studies that were published a few years ago by a spectacular MD PhD student in the laboratory, Wilfried Pastor, who among his many, many gifts was the ability to reach out and collaborate with people. And this is something I'd like to transmit to people generally. The more you reach out and form networks, the more you have a chance to make real progress. So what Will did was basically establish the system that had been pioneered by Mina Bissell, who many years ago discovered that if you grow cells in a more natural setting where they're actually implanted in laminin-rich extracellular matrix, which replicates the way the cells in our body are present. They're not just growing on plastic in a dish. They actually form structures that, in the case of mammary cells, replicate to a first approximation the terminal duct of the mammary gland. As the cells go through the process of growing in this medium, they form spheres. The spheres get hollowed out. And in fact, if they're really normal cells and you give them certain hormones, they actually secrete milk proteins into the lumen there. And what Mina had also discovered, and this is the next slide, was that's true of normal cells, but if you take cells from any breast cancer, they don't do that. They form disordered, irregular clusters in the same setting. And one cell line, however, that we worked with derived from a breast cancer, known as MDA468, a tiny fraction of the cells actually did form these normal, what we call acinar-like structures. But oops, what did I do here? That's horrible. Okay, go back. All right, so a colleague of mine, Jill Barganetti, who was a long time ago a postdoc in my lab, actually engineered these cells so that you could reduce dramatically the level of P53 in those cells, knock it down. And in the process, that's how you do it with doxycycline. We'll put these cells into the same protocol, and instead of having a I'm sorry, I have to learn how to, I'm a little dyslexic here. Okay, so instead of having a tiny fraction of these normal structures knock down mutant P53, over half of the clusters in this experiment actually look normal. So that indicated, and we proved essentially, that P53 was maintaining the malignant characteristics of those cells in that setting. So that caused us to do a gene expression analysis and a pathway analysis to try to figure out what was the difference between having high P53 and low P53. And it turned out that several pathways were altered. And the one that really caught our eye, the one that was the most significantly affected, was a pathway I never would have considered thinking about even, which was the pathway by which cells synthesize cholesterol. So, and this is seen here, it's called the mevalonic pathway, and I won't go through all the experiments that Will did to validate that this was the case, but this is going to be now the theme of this talk, why we believe this pathway is very critical and important and P53's regulation of it, or loss of P53's deregulation of it, we believe is very critical for cancer. So this pathway, again, this is what the pathway looks like. It not only synthesizes cholesterol, but many of the byproducts along this way are very critical for many other aspects in cells. And in fact, this pathway has been the target of several kinds of drugs that affect cancer. Most famous of these are statins. I'm sure many of you have heard of them. They're drugs they're blockbuster drugs that are used to treat hypercholesterolemia. They're taken by many people. I don't know the numbers, but it's in the millions and millions in this country. They're very effective. There are many other drugs too. I don't think I have it on this slide that have been developed to affect other aspects of this pathway, such as protein prenylation. So, here they are over here. So what I wanted to go on with this, first of all, very quickly, we show that if you actually take different breast cancer cell lines that have mutant p53 in culture and treat them with statins. Basically, the statins are incredibly effective, and they're not that effective. In fact, they're virtually ineffective when we do the same thing with normal cells. These cells are somehow dependent on the cholesterol biosynthesis pathway for survival, for viability. And I think this is an important thing to think about in terms of potential cancer treatment. Not only that, but Will was able to show that the target of these drugs, namely the ability causing the cells to not be able to form a very critical precursor in this pathway, mevalonic acid. If you put mevalonic acid into these cultures, not only do you prevent the statins, but you prevent this morphological reversion to normal looking like structures in cells. So that meant that the reason why mutant p53 is maintaining the malignant characteristics of these cells is because of its ability to activate that pathway. So I'll go very briefly through this because I'll be coming back to it in a few minutes. This pathway it's basically the Nobel Prize winning work of Joseph Goldstein and Michael Brown a few years ago. And among other things, they figured out how it's regulated. It's regulated by a single transcription factor called SREBP2. And SREBP2 basically, we showed interacts with mutant p53, and mutant p53 stimulates the ability of SREBP2 to activate these genes and thereby to increase the malignant morphology of these breast cancer cells in cultures. And we also collaborated with Annalisa Borison-Dale in Norway, who had several data sets from breast cancer patients. And with her, we discovered that that the cluster of patients that had the highest number or proportion of mutant p53 also had the highest level of all of these enzymes that are involved in the cholesterol biosynthesis pathway. And this correlated in turn with poorer survival of these breast cancer patients. So those who had the highest levels of these enzymes also had worse survival. So this is something to think about as I've been listening to this. And it would be very interesting to look at cells from tumors of Li-Fraumeni patients to see whether they have unusually high cholesterol biosynthesis enzymes, because I think that could be a vulnerability that we should think about in the future. All right, so I want to move on now, and I'm going to go through this part quite quickly because I want to get back to the cholesterol biosynthesis pathway. But I think we did learn something as well from the well of another wonderful MD PhD student in the lab, Neil Pfister. He just left a little while ago. He was very interested in looking again for another genes that mutant p53 might regulate, and he discovered a very interesting gene that plays I think an important role in cancer biology in general. It's the receptor for a ligand called VEGF, which in turn is necessary for stimulating the synthesis of new blood cells. So for those of you who think about cancer and tumors, a tumor never grows as an isolated ball of cells. It has to interact extensively with the cells and the tissues around it. And most importantly, it needs to ensure that it gets its own blood supply. So one of the things that tumors always do is produce, induce the surrounding tissues to form blood vessels. And so it turns out mutant p53 can be involved in this. Mutant p53 can induce VEGF-R2, the receptor for VEGF. It can also actually work. Some cells can actually get changed. Some tumor cells evolve to the point where they not only secrete the hormone, they also secrete the receptor. That's what we call autocrine signaling. And that means that they can basically stimulate their own growth that way. And it turns out the breast cancer cells we work with did show that kind of autocrine signaling. And I won't get into it in great detail, other than to show that Neil worked in very deeply to try to understand how mutant p53 is regulating this particular gene. He showed that it actually causes the chromatin in the vicinity of the initiation point for making the RNA of this gene to be displaced. And he showed that mutant p53 can interact with a very specific complex called the SWI/SNF chromatin remodeling complex. And in doing so, it actually stimulated transcription of this gene. He did a lot of experiments to support this, which I won't show this now. But this just gives you a little information about this. It turns out that these complexes on cells, they don't only work for this specific gene, they actually help to remodel, as we call the chromatin, around maybe a third to a fourth of the genes in our cells. And they are very complex. There's a very active field studying them. It turns out that the SWI/SNF complex genes in it tend to be tumor suppressors. So some of them are lost in many human tumors. But interestingly, there is what you call a reciprocal or exclusionary relationship between tumors that have mutant p53 and tumors that have mutations in the SWI/SNF. So it's as if mutant p53 needs to work with SWI/SNF in order to activate not only VEGF, but a goodly number of its other genes. And in fact, we did RNA-seq analysis where we knocked down either mutant p53 or these key components of the SWI/SNF. These are the enzymatic activity genes in this or proteins in this complex, and we found that mutant p53, over 50% of all the genes that mutant p53 regulates. And by the way, when you modulate the levels of mutant p53 in breast cancer cells, you'll see about 1,000 genes being affected. Half of them go up and half of them go down. Mutant p53 is essentially regulating what we call the transcriptome of the gene, and that's how we ended up figuring out the cholesterol biosynthesis. Once you get all the information from these genes, there's various programs for actually analyzing the pathways that they're regulating. And so it turns out that a good number of the genes that mutant p53 regulates requires this complex. And this is the hypothesis that Neil developed, that mutant p53 interacts with this complex and stimulates remodeling of the complex. And he produced that very strong evidence for this with respect to this particular gene he worked with, and we believe it has effects on other genes as well. In fact, very recently, we've done the exact same analysis now looking at the genes that regulate the cholesterol biosynthesis pathway, and we're getting essentially the same result. You knock down these key components of SWI/SNF, and you get much less activity of these different genes that we've looked at over here. These are just an example of 3 of them. So this is just new data, hot off the press, which we're still working on. So now I'm going to move on for the last part to what we think is perhaps the most, first of all, it's unpublished, so I'm very excited about it. And second of all, I think it really solidifies the relationship between p53 and what we call the mevalonic pathway, or the pathway by which cells synthesize cholesterol and other products. The take-home message essentially is it's not only that mutant p53 can upregulate this pathway, but you need wild-type p53 in your cells to restrain it. And here's some of the evidence for that. So one of the things I want to point out is this fascinating kind of what we call yin-yang relationship between wild-type and mutant p53. Very often genes and pathways that are upregulated by wild-type p53 are reduced in cells with mutant p53, and conversely, very often genes that we have found to be, or pathways that we've found to be actively increased and stimulated by mutant p53 are actively repressed by wild-type p53. So this is the work of a postdoc in the lab who's really a deep thinker and a wonderful experimentalist, Sung-Won Moon. And he's also we've been really lucky to have a great collaboration with Scott Lowe and his group, with particularly Chun-Hao Wang, a spectacular graduate student in his lab. We have a grant together with Scott and a few other people in the New York area, and if we hadn't had a multi-lab meeting, which we do from time to time, and Sungwon hadn't presented his work, I wouldn't be able to provide to you really exciting evidence in mice, that in mouse model, that the mevalonic pathway needs to be restrained in order for liver tumors to form. So this is just some basic data. What Sungwon did was to show that if you have wild type p53 in a specific cell line, whoops, I went backward, a specific cell line, and you induce p53 with a very specific drug called nutlin. It induces wild type but not mutant p53. You get many of these genes are significantly reduced. You don't see this in the cell lines, the exact same cell line from which p53 has been deleted. It's a more physiological way to do this is to activate the mevalonic pathway by removing cholesterol from the cells. And if you do that and you compare cells with wild type versus loss of p53, you get much more activity in the p53 null cells against different mevalonic pathway genes. And this master transcription factor, which I'm now going to turn to for a minute, the SREBP2, binds much more efficiently to the promoter of its genes, such as the HMG-CoA reductase gene. You don't have to know what that is, when cells that don't have p53. So this just explains, and again, this is a very beautiful system that was, regulatory system. So if you have enough cholesterol in your cells don't want to make cholesterol. That's a normal thing. However, if they're deprived of cholesterol, this activates a series of steps that result in a very large protein being moved from one part of the cell to another, where there are two proteases that cut proteins, waiting to cut this particular large protein and release from it this transcription factor known as SREB2. So this is the precursor, and this is the mature protein over here. And I should be pointing here too, it's not fair. You can see, therefore, that it all depends on the levels of cholesterol in cells. So you remove cholesterol, you activate this whole process. High cholesterol, you prevent it, unless p53 isn't there. So what we discovered is that wild-type p53 is repressing this ability to undergo maturation. And this is the mature form over here. And if you see there's much more, you see there's much more of the, this mature form in cells that don't have p53 than cells that do. And we've shown that in a couple of other cell lines as well. These are liver cells. These are cell lines where we've artificially put p53 into them. Now you lose the mature form here, or here if you activate p53 in these cells with wild type, you get much less of this mature form, which means you get much less synthesis of these different enzymes in this expression of these enzymes in this system. This is really a sort of an area I won't get into, but p53's, one of its main target genes, is essential for this, and we still don't fully understand the mechanism, but it's something that we're working on very hard. What I would like to say, however, is, oh yes, and there's another interesting thing. If we take cells and follow them progressing through the cell cycle, after you've removed sterols from the media, you see that if you don't have p53, they move much more effectively through the cell cycle than the cells with wild-type p53, and this correlates with having a much higher level of the mature form of this particular transcription factor in the absence of p53. So this is where our collaboration with Scott Lowe and his lab comes in. Chun-Hao did an analysis of, I'd say, almost 500 different cell lines, separated them into wild type or mutant. Mutant simply means it doesn't have wild type. It could have not wild type because it lacks the protein, or it could have one of these missense mutations. And basically his analysis showed that there's overall far lower levels of these enzymes in aggregate in the wild type p53 containing cell lines than in any of the cell lines that have mutant p53. So we started to work, or he did this work in mice, working with the engine that is Scott Lowe's lab. He's got very high-tech mouse models, one of which involves injecting into the tail of the mouse an oncogene, MYC, which many of you may have heard of, along with constructs that are known as CRISPR constructs. They destroy the ability via gene editing to express any p53 at all. And if they're injected into the tail of the mice by a process called hydrodynamic tail vein injection, they go through the system into the liver. And within 4 to 5 weeks, the mice get hepatocellular carcinomas. This is an amazing acceleration of the speed of doing research. In the normal way that people generate these mouse models, it takes between 1 to 2 years. And so the results we've discovered are as follows. First of all, you see much higher levels of mature p53 in tumors that were generated with another tumor suppressor but still have wild-type p53 than in the tumors with mutant p53. And then, perhaps most excitingly, what Chun Hao did was use the same approach now to get rid of enzymes, the product of the genes that are in the mevalonic pathway. And the results were really very dramatic. These are not, it's always kind of disturbing to look at tumors, but at least the results were very exciting. If you used a control shRNA, which was used to ablate these things, got lots of tumors, no tumors formed at all when you ablated any of the genes that he used in the cholesterol biosynthesis pathway. And this was not the case in tumors that retained wild-type p53, but were generated by another tumor suppressor. In fact, when he treated these mice with statins, and that's sort of the bottom line here, got the same result. You could reduce dramatically the size and the frequency of the tumors if they lacked p53, but not if they, but not if they were generated by this other mechanism where they still had wild-type p53. So this is our hypothesis, essentially, that, for example, with the mevalonic pathway, and this is basically the last couple of slides I'm going to show, wild-type p53 actively shuts down or dampens this pathway. If you don't have that mechanism, it's not only that the levels of the products of the enzymes and the whole ability to synthesize cholesterol and its byproducts go up, but the cells depend on it. They actually become dependent. And that's a vulnerability that I believe could be exploited let's say we call this the era of personalized medicine. If you could actually assess if a patient's tumors have high levels of these enzymes, and it would be very easy to do that, I would say statin treatment might be a very promising way to look at it. And then my hypothesis goes a little further that says if you have mutant p53, the levels are still even higher. Whether that's the case or not, we have some evidence in, I won't get into this, it's not worth it, but we have a little bit of evidence that we have very clear evidence. We've looked through many of these genes, comparing wild type versus any mutation in cells. There's always lower levels of these enzymes, or very frequently. And then in some cases, with some of the genes, it's still higher when you have a real mutant p53 protein. So this is the summary of what I've told you before. The mevalonic pathway, I think, is regulated both up and by mutant p53, down by wild type p53. We've seen this in several types. In breast cancer cell lines with mutant p53, it not only upregulates this pathway, but this correlates actually with patients' tumors and survival. And our studies in 3D cultures in mice reveal a dependency on this process that I believe could be exploited. And there is, in fact, good clinical data already in some cancers that statins have had a beneficial effect. So I think it's something the Li-Fraumeni community might think about as well in the future. And this is just my group right now, very international, as is the case with many of our different groups. Many of the people whose work I've showed are no longer here. This is Sung-Wan over here, who did the most recent work, and these are the rest of the lab. Thank you.